Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Mineral nutrition and solute transport in plants. Table of contents. Introduction to mineral nutrition and solute transport in plants. Macronutrients and micronutrients. Functions and roles. Mechanism of mineral uptake in plants. Function of root hairs in absorbing nutrients. Plant transport systems, Xylem and Phloem. Translocation of solutes. Factors that influence mineral nutrition and transport. Deficiency symptoms of essential nutrients. Soil pH and how it affects nutrient availability. Mycorrhizal associations and nutrient uptake. Techniques to enhance nutrient use efficiency. Environmental stress and how it affects nutrient. Transport. New advances in fertilizer application for improved nutrition. Conclusion. Introduction to mineral nutrition and solute transport in plants. Plant growth and development rely heavily on mineral nutrition as a fundamental requirement. Mineral nutrients are absorbed from the soil in ionic form, and each nutrient performs specific biochemical and physiological functions within the plant. Among the 17 known essential elements, some are required in large quantities and are categorized as macronutrients, while others are needed only in trace amounts and are categorized as micronutrients. All essential elements, regardless of whether they are required in large or small amounts, are equally important and cannot be substituted by any other element. A deficiency of even one essential nutrient can significantly disrupt plant metabolism, affect structural integrity, or impair reproductive processes. These mineral nutrients play key roles in photosynthesis, enzyme activation, protein synthesis, and energy transfer within the plant system. Understanding the nature of these nutrients, along with their roles and mechanisms of absorption, is important for promoting sustainable agriculture and improving overall crop productivity. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Macronutrients and micronutrients. Functions and roles. Both macronutrients and micronutrients are essential for plant growth, differing only in the quantities required, not in their importance. Macronutrients. Macronutrients including nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur are needed in relatively large amounts to support vital physiological processes. Nitrogen plays a key role in the formation of amino acids and proteins, which are fundamental to plant structure and metabolism. Phosphorus is necessary for energy metabolism and is an integral part of genetic material, contributing to cellular energy transfer and DNA/RNA structure. Potassium is involved in enzyme activation and osmotic regulation, helping maintain water balance and supporting various metabolic reactions. Calcium strengthens cell walls and contributes to structural stability throughout the plant. Magnesium is essential for the development of chlorophyll, making it vital for photosynthesis and energy capture. Sulfur is required for the formation of certain amino acids and vitamins, contributing to protein structure and plant health. Micronutrients. Micronutrients, including iron, manganese, boron, zinc, Copper, molybdenum, and chlorine are required only in small quantities, but are equally critical for overall plant function. Iron is essential for chlorophyll synthesis and participates in electron transfer during photosynthesis and respiration. Manganese contributes to photosynthesis, especially in the water-splitting reaction of photosystem II. Boron maintains cell wall integrity and supports pollen tube elongation, influencing reproductive success. Zinc plays a role in hormone regulation and is involved in enzyme activation and protein synthesis. Copper functions as a component of several oxidase enzymes necessary for redox reactions and metabolic activity. Molybdenum aids in nitrogen assimilation by supporting nitrate reduction and nitrogen fixation. Chlorine regulates stomatal opening and closing, which affects water balance and gas exchange. Balanced availability of both macro and micronutrients is vital to ensure optimal plant metabolism, enhanced disease resistance, and proper reproductive development. Mechanism of mineral uptake in plants. Mineral nutrient absorption from the soil is a highly regulated process that often requires energy. Plants use two major mechanisms for nutrient entry into root cells: passive transport and active transport. Together, these mechanisms ensure efficient acquisition of a wide range of nutrients, even when soil nutrient levels are low. Passive transport. Passive transport does not require energy and occurs along the concentration gradient. Ions move into root cells either by directly permeating the cell membrane or by passing through protein channels. Since no ATP is used, this method is slower compared to active transport. Passive transport mainly accounts for the initial movement of ions from the soil solution into the outer root cells. Active transport. Active transport requires the expenditure of ATP to move ions against their concentration gradient. Specialized transporter proteins in the plasma membrane facilitate selective uptake of nutrients, especially when external concentrations are low. ATP-driven pumps, such as H+ ATPase, create electrochemical gradients that assist in the absorption of charged ions. This mechanism provides plants with the ability to accumulate essential minerals even in nutrient-poor soils. Pathways for internal movement of nutrients. After entering the root, minerals must travel through internal tissues to reach the xylem, and this occurs through two main pathways. Apoplastic pathway. In the apoplastic pathway, water and dissolved minerals move through cell walls and intercellular spaces without crossing the plasma membrane. This pathway allows relatively rapid movement of substances. Transport through the apoplast is blocked at the endodermis by the Casparian strip, a suberized barrier that forces substances to shift into the symplast for selective entry. Symplastic pathway. The symplastic pathway involves the diffusion of minerals through the cytoplasm of root cells. Movement occurs from cell to cell via plasmodesmata, which are cytoplasmic connections between adjacent cells. Minerals must first cross the plasma membrane to enter the symplast, ensuring selective uptake. Once inside, they can move freely through linked cytoplasmic channels toward the vascular tissues. This pathway provides stricter regulation over which substances enter the xylem. Overall integration. The combined function of passive and active transport ensures effective nutrient uptake from the soil. The apoplastic and symplastic pathways coordinate to deliver and regulate mineral movement toward the xylem. Together, they ensure efficient, controlled, and selective mineral transport before nutrients are finally loaded into the xylem for long-distance movement throughout the plant. Function of root hairs in absorbing nutrients. Root hairs are elongated, tubular extensions of root epidermal cells and serve as the primary sites for the absorption of water and mineral ions. They greatly increase the surface area of the root system, allowing the plant to explore a larger volume of soil and acquire nutrients more efficiently. Root hairs are located near the root tips, where active growth occurs, and they extend between soil particles to maximize contact with soil water and dissolved minerals. These structures play a crucial role in the initial stages of nutrient uptake through both passive and active transport mechanisms. Their direct exposure to the soil solution enables the absorption of essential ions such as nitrate, phosphate, potassium, calcium, and magnesium. The presence of thin cell walls and a lack of a cuticle allow minerals to diffuse easily into the cells. Root hairs also form interactions with beneficial soil microorganisms and mycorrhizal fungi, which enhance nutrient availability and support improved mineral uptake. Download our microbiology study app made for researchers like you. Plant transport systems, Xylem and Phloem. Plants rely on two highly specialized vascular tissues, xylem and phloem, for internal transport of essential substances. The xylem is responsible for the upward movement of water and dissolved minerals from the roots to the shoots. This upward transport is mainly driven by transpiration, a process in which water evaporates from leaf surfaces, creating a pressure deficit that pulls water upward through xylem vessels. Xylem tissue also provides mechanical support to the plant due to its thick, lignified cell walls. The phloem transports organic nutrients, especially sugars like sucrose, in a bidirectional manner depending on the plant's needs. Movement in the phloem occurs from source tissues, such as leaves, to sink tissues like roots, fruits, and developing organs. Translocation through the phloem requires energy and involves living cells, including sieve tube elements and companion cells. The coordinated functioning of both xylem and phloem ensures efficient distribution of water, minerals, and food, supporting overall plant growth, development, and reproduction. Translocation of solutes. Translocation refers to the movement of organic solutes, mainly carbohydrates, through the phloem from their production sites, sources, to their utilization or storage sites, sinks. In most plants, mature leaves act as source tissues because they conduct photosynthesis and produce sugars. These sugars are actively loaded into the phloem and transported through sieve tubes under pressure towards sink tissues such as roots, young leaves, flowers, seeds, and storage organs. The mechanism driving this movement is explained by the pressure flow hypothesis. At the source, sugar loading into the phloem increases the osmotic pressure within the sieve tubes, causing water to enter from the nearby xylem and raising turgor pressure. At the sink, sugars are unloaded, decreasing osmotic pressure, which allows water to exit back into the xylem. The difference in turgor pressure between source and sink generates the force that pushes the phloem sap along the sieve tubes. This process is highly regulated and essential for distributing energy, supporting reproductive growth, and enabling plants to respond effectively to environmental changes. Factors that influence mineral nutrition and transport. Plant mineral nutrition and the efficiency of nutrient transport are influenced by multiple internal and external factors. Soil characteristics such as structure, texture, organic matter content, and mineral composition directly determine nutrient availability to plants. Soil moisture plays a key role in governing the solubility and mobility of ions, affecting how easily roots can absorb them. Adequate soil aeration is essential as it supports root respiration, which in turn influences the uptake of mineral nutrients. Environmental factors including temperature, light intensity, and humidity affect root metabolism, transpiration rates, and the activity of enzymes involved in nutrient transport. Internal plant factors such as growth stage, root architecture, membrane transport proteins, and metabolic demands regulate nutrient absorption, movement, and utilization. Symbiotic associations with nitrogen-fixing bacteria and mycorrhizal fungi also modify nutrient dynamics by improving nutrient availability and enhancing plant uptake. Understanding these influencing factors is important for developing effective fertilization practices and ensuring improved plant health and crop productivity. Deficiency symptoms of essential nutrients. When plants lack certain essential nutrients, they develop characteristic deficiency symptoms that negatively affect growth, development, and yield. The appearance of symptoms depends largely on the mobility of the nutrient within the plant. Mobile nutrients such as nitrogen and magnesium show deficiency symptoms first in older leaves because these nutrients move easily to younger tissues. Immobile nutrients such as iron and calcium show symptoms in younger tissues since these nutrients cannot be readily transported from older parts of the plant. Nitrogen deficiency results in chlorosis, yellowing, of older leaves and leads to overall dwarfing of the plant. Phosphorus deficiency causes dark green or purplish discoloration, especially noticeable in leaves and stems. Potassium deficiency leads to yellowing along the leaf margins and causes stems to become weak or slender. Calcium deficiency may result in blossom end rot in fruits and poor root development. Iron deficiency appears as intervenal chlorosis, primarily affecting young leaves. Early detection of these deficiency symptoms is crucial so that plants can be corrected through targeted fertilization or appropriate soil amendments. Soil pH and how it affects nutrient availability. Soil pH is a key factor that determines the availability of nutrients to plants. Most essential nutrients are readily available to plants when soil pH ranges from 6.0 to 7.5, which is slightly acidic to neutral. Outside this optimal range, certain nutrients may become insoluble or even reach toxic levels. In acidic soils, low pH, metals such as aluminum, manganese, and iron can accumulate to toxic concentrations, while essential nutrients like phosphorus, calcium, and magnesium become less available. In alkaline soils, high pH, the availability of micronutrients such as iron, zinc, copper, and manganese decreases, often leading to deficiency symptoms. soil pH also affects the activity of soil microorganisms that are responsible for nutrient cycling. Regular soil testing and pH adjustment by adding lime to raise pH or sulfur to lower pH can improve nutrient availability and promote healthy plant growth. Mycorrhizal associations and nutrient uptake. Mycorrhizae are symbiotic associations between plant roots and specific soil fungi. Fungi colonize the root surface and extend hyphae into the soil, greatly increasing the effective surface area for nutrient absorption. In exchange for sugars supplied by the plant, mycorrhizal fungi enhance the uptake of relatively immobile nutrients, such as phosphorus, zinc, and copper. There are two major types of mycorrhizal associations. Ectomycorrhizae form a sheath around roots and are commonly found in trees. Arbuscular mycorrhizae, AM, penetrate root cells and are widespread in most crop plants. Mycorrhizal associations improve plant tolerance to stress, enhance soil structure, and contribute to long-term soil fertility. These associations also reduce reliance on synthetic fertilizers, making them important for sustainable agricultural practices. Techniques to enhance nutrient use efficiency. Improving nutrient use efficiency, NUE, is critical for maximizing crop productivity while reducing negative environmental impacts. Balanced fertilization. Applying nutrients in the correct ratios based on soil testing prevents deficiencies and avoids toxicities. Split applications. Dividing fertilizer doses into multiple applications reduces nutrient loss through leaching or volatilization. Slow-release fertilizers. Using formulations that release nutrients gradually over time ensures steady availability for plant uptake. Foliar feeding. Direct application of nutrients to leaves helps quickly correct deficiencies and supports plant growth. Soil amendments. Adding organic matter, biochar, or beneficial microbes improves nutrient retention, enhances root health, and promotes soil fertility. Precision agriculture. Utilizing GPS and sensor technologies allows for site-specific nutrient management, optimizing inputs, and reducing waste. Crop rotation and intercropping. These practices maintain or improve soil fertility and prevent nutrient depletion. Implementing these strategies enables farmers to achieve higher yields, lower input costs, and reduced environmental degradation. Environmental stress and how it affects nutrient transport. Plants are frequently exposed to environmental stresses such as drought, salinity, extreme temperatures, waterlogging, and heavy metal pollution, all of which significantly affect nutrient uptake, transport, and utilization. Drought stress reduces soil water availability, limiting nutrient solubility and root growth. Lowers transpiration rates, which decreases passive transport of nutrients like nitrogen and calcium. Salinity stress introduces excess sodium and chloride ions, interfering with the absorption of potassium, magnesium, and nitrate. Disturbs ion balance and creates osmotic stress in plants. Temperature stress, extremely high or low. Temperatures alter root metabolism and membrane fluidity, reduces efficiency of active nutrient transport processes. Flooding or waterlogging decreases oxygen availability in the soil, limiting root respiration, restricts energy supply required for active nutrient uptake. Plants respond to environmental stress through adaptive mechanisms, including increased expression of stress-associated transporters. production of compatible solutes to maintain cellular function, symbiotic associations with stress-tolerant mycorrhizal fungi. Understanding these physiological responses is crucial for crop breeding and management strategies aimed at improving tolerance to climate change. New advances in fertilizer application for improved nutrition. Several innovative methods have been developed to enhance nutrient use efficiency while reducing environmental impact. Nanofertilizers supply nutrients in the form of nanoparticles, improving uptake efficiency and minimizing nutrient losses. Controlled release fertilizers, composed of coated pellets that release nutrients gradually, matching plant growth requirements over time. Biofertilizers utilize beneficial microorganisms such as Rhizobium, Azotobacter, or phosphate-solubilizing bacteria to naturally enhance nutrient availability. Fertigation integrates fertilizers with irrigation systems to ensure even nutrient distribution and higher uptake efficiency. Application of AI and drones employs aerial technology and predictive analytics to deliver nutrients accurately and timely at the field level. These advanced techniques not only boost crop yields, but also help conserve water resources and maintain soil health. Conclusion: mineral nutrition is a fundamental pillar of plant physiology and a key component of sustainable agriculture. Proper plant nutrition encompasses understanding the essential nutrients, their forms, mechanisms of uptake, and internal transport. Nutrient availability and transport are influenced by both environmental conditions and soil characteristics, making plant nutrition management a complex but critical task. Advances in fertilizer technologies, including nanofertilizers, controlled release fertilizers, and biofertilizers, offer improved nutrient efficiency. Insights into root microbe interactions, such as mycorrhizal associations, enhance nutrient uptake and soil health. Precision farming technologies, including AI and drones, allow targeted nutrient management to optimize efficiency. Combining these approaches enables maximized crop productivity while maintaining ecological balance and sustainability. Through the arrangement of scientific information and sustainable technologies, we can satisfy increasing food production needs while protecting natural resources and enhancing climate-resilient cropping systems. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!